Social insects are a peculiar minority, about 2% of species of insects, that characteristically live in structured, durable groups that are known as colonies. The key to, or a key to understanding social insects is the, go to the next slide please, is the concept of caste, which has to do with the different colony maintaining roles of different kinds of individuals in the colony. And the primary distinction is between reproductive individuals, such as this queen here, and non-reproductive workers. Social insects are a conspicuous part of our environment, and you're all certainly well familiar with them. Now, I'm going to treat a fairly large theme in the biology of social insects. I'm going to treat it a little more technically than many of our lectures. You're going to hear some new terms. It's going to be data rich. However, unlike, for instance, at the university very often, if I'm listening to a lecture in a subject outside of my specialty, then I follow it for about 10 minutes and then I get lost. And I'm just listening to the music of it and absorbing the flavor without really knowing what's going on. That's not going to happen. I guarantee that you are going to follow me every step of the way. I'm going to make sure that you understand everything I say. So I don't want to see the eyes glazing over. It's completely unnecessary. So you're going to come away from this lecture knowing something important about social insects, whether you wanted to or not, which I expect you do. Now, you know what social insects are. Let's make sure we know who they are. There are 4 great groups. Now, most wasps are solitary, such as the ones that made these nests here. And you've seen these, black shiny wasps, about the size of a Jack Spaniard, but slimmer, and they make these bud nests, and they're solitary. They don't live in groups. However, social wasps include this one. Now, this one is named after, this is Miscociferous baconi, named after the professor of zoology. And as it gets bigger, there will be a lot of individuals, both brood and adults, on the nest. And here's another one. This is the previous one we would call in Trinidad a Jack Spaniard. And this one we would call a Maribona. And you notice that although they're still social wasps, there are many more of them. The colonies are much bigger. You would expect them to be socially more complex, and you'd be right about that. And although you notice the nest is covered, and there's a whole bunch of cones inside, another kind of social wasp. Now, the most familiar of the social bees is the western honeybee, which is now common here, but it's not native to here. We also, while we're at it, so here's a Pete. What's this say? Pete, give us a translation. Oh, you're too slow, Peter. Worker, drone, and last chance.
Queen.
Queen. Okay. All right. Okay, so let's. Here is a nest of a stingless bee. Stingless bees are native to this part of the world. There's one nest, and this is part of the Mona nigrior, and you've seen them. This thing, it kind of looks like a cow pie on branches. Sometimes you get more than yes, please. You get more than one on a building or in a particular place, and these have a social structure comparable to that of honeybees. Fairly complex, fairly large colonies. Now, most bees, like most wasps, are solitary. The social species are in the minority. However, ants, like this one, the one that built this nest, and you'll occasionally see these if you're attentive in Arena. That's the only place I've seen them commonly. All ants are social. They are presumably derived from solitary ancestors, but they themselves are not solitary. And here's another one that built this. Look at those 2 stalwarts. And this is, of course, our beloved Bachak. They built this great big mound there, and inside, even though very few people have taken the trouble to excavate one and actually count the individuals, you would expect there to be hundreds of thousands of individuals in there, and you'd be right. And you'd expect, therefore, that there would be a fairly finely divided division of labor, specialization of labor among the castes. And once again, you'd be right. Now, these three, the wasps, the bees, and the ants, are all part of the order Hymenoptera. So they're sort of closely related, not especially closely related. The other group, the termites, and this is Pseudotermes ornager. You've all seen this nest, okay? And you break apart part of it, you see the termites inside. These are, in fact, now known to be very closely related to cockroaches. And they can, in fact, be seen as social cockroaches. And if you open up the nest, here's what you see. Now, one peculiarity of termites is that most species have a highly special, have a non-reproductive caste specialized for defense of the colony, and they're known as soldiers. So you open this up, and you see the workers, the ordinary workers, retreat, the soldiers come out, and it kind of looks like a squirt ball, doesn't it? Well, these have chemical defenses. And so if you smell it and it smells kind of like pine pitch, well, those are terpenoids in there. Similar defensive compounds to what pine trees have. Okay, now, in many respects, a social insect colony resembles, in its organization, a single multicellular organism. So that there are, let's see, Chris, can we do something about the light so I can just, we don't want to take it up very much, but if you can just give it.
There's no adjustment for on off.
Okay, let's take it off. Okay. So, in many respects, a colony resembles a single organism. It's like an organism of organisms, an organism at a higher level, or a so-called superorganism. So, for instance, the different castes with their different tasks, different colony-maintaining tasks, are sort of analogous to organ systems in your own body or in another body. There are functions that are comparable. For instance, yes, please. Now here you see this honeybee here is fanning at the nest entrance. She's creating a current. Well, you know that your own body works best within a fairly narrowly defined range of temperature. And so you maintain that body temperature. If you go to the doctor takes your temperature right away. And if it's more than slightly off of the normal temperature, then that's an indication that there's something wrong. Okay? Similarly, many social insects maintain a fairly constant temperature within the nest. And this, as in your body, is through negative feedback mechanisms. Similarly, you all know what fever is. People think of fever as being a sickness. Well, not really. Fever is a response to a sickness. Fever is your body's way of heating up in order to try to kill something that you've got in you. So, if you've got a fever, you're happy when the fever's over, but the doctor may not take measures to lower the fever, because that might not be good for you. So some very clever entomologists said, well, sometimes honeybees also have, okay, thank you. Oh yeah, love technology. Sometimes honeybees also have disease organisms that are heat sensitive. So they predicted that honeybees would be found to have fever at the colony level, not at the single organism level. And so using this superorganism concept as a basis for predictions, they experimentally tested that and found that, in fact, there is fever at the colony level. An especially striking way that the colony resembles an organism is in colony cycles. And now we're coming, now we're coming to the subject matter for today. This is analogous to an organism's life cycle. Birth, growing up, maturation, reproduction, senescence, eventually death. Colonies go through a fairly predictable, orderly cycle that is similar to this. So, for instance, on your handout here, which you can't see, but now I can, you've got, let's go to it, please. Okay, so that's what you've got there. This is the colony cycle of a Jack Spaniard wasp, genus Colonies. And if this graphic representation of it doesn't really work for you, don't worry about it. I'm going to go through it. I'm going to narrate it as well. So the beginning of the colony cycle happens right up here, where a queen or a group of queens founds a colony. And here are the brood in the outer circle. Here are adults in the inner circle. And they build a nest, not shown there. And they start producing brood. They lay eggs and start rearing them up. And there comes a time, seen right there, when the first adult individuals emerge. And these are workers, always workers. So that stage, from the very beginning down to here, is known as the founding stage. It is fairly distinct in what the colony does and what it's all about. After that, more and more workers are produced. The workers produce more workers, lots more brood. The queen lays eggs, but no longer goes out foraging. And then there comes a time when the queen starts laying eggs that will turn into males and new queens. And in time, these emerge. And then somewhat later, they fly off, or the males fly out. They mate if they're lucky. The new queens mate, and they start new colonies, and it starts over again, and the colony disintegrates. Okay? The end of the colony. Now,
that's the original colony.
That's what?
The original colony.
Yes.
Not that.
Yes, that's right. Leaving the nest, and we're going to come back to that. So the nest remains, as you've seen, I'm sure. Okay? So, founding stage, growth stage, reproductive stage. Three fairly distinct stages with clear beginnings and ends in this cycle. Now, on the second page, on the flip side of your handout, you see the approximate numbers of species of social insects in the four great groups, and you see that there are thousands and thousands of species. So you might reasonably expect that there would be many different kinds of colony cycles, and you'd be right about that. However, the colony cycles can be categorized with confidence into four basic patterns. And here they are. Now, there's the one we've just seen up there. So virtually every species of social insect in the world can be put with very little distortion into one or another of these four patterns. Now, if the graphic representation confuses you a bit, don't worry, I'm going to cook it down. Because these have two basic parameters. There are two basic variables in this that account for the four patterns. So, the colony can be founded by nothing but a reproductive or a group of reproductives without the aid of workers, as in Colonies, as in the Jack Spaniards that we've just seen, so that there are no workers until the end of the founding stage, or the colony can be founded by a group of workers and reproductives together. So this latter is known as swarm founding. It's in your glossary. Swarm founding. A good example of a swarm founder is honeybees. So you know that when they start out, when the honeybee colony starts out, it's never without workers. The queen cannot do anything without workers. She doesn't have the apparatus to, she doesn't have pollen baskets. She doesn't have the behavioral or physical apparatus to form a nest. So that's one parameter. Independent founding of new colonies, as in Jack Spaniard wasps, versus swarm founding. The other one has to do with whether the colony has just one reproductive episode or many. Now we think of plants, for instance. The herbaceous plants in your garden mostly just grow up, they go to seed, and then they die. So that they've just got one reproductive episode. On the other hand, the trees in your garden, typically they can reproduce over and over and over again. And if they produce a really big crop of fruits, as my Portugal trees have done this year, next year that takes it out. Reproduction tends to be a fairly draining process. So my ex-wives have told me. And so my Portugal trees will probably not have much of a crop next year, no matter what the conditions are. But they will reproduce again. Now there are some big, and most big animals are like that. Small animals, most insects just reproduce once. Now there's some exceptions. So you think of Pacific salmon. Pacific salmon have just one reproductive episode, and then the Lord calls them home. Or you think of the talipot palm and many bamboos. They grow for years and years and years. They don't reproduce, and then suddenly the talipot palm has this great big gorgeous flowering, produces a lot of propagules, and then it dies. Whenever you see a talipot palm in flower or in fruit, you know it's not going to last very long. So that is the other the other parameter. So up there we see the ones with what is called a determinate colony cycle. One reproductive episode, and then the colony dies. And now here we see the ones with an indeterminate colony cycle. So for instance, with swarm founding and indeterminate colony cycle, we've got stingless bees and honeybees. With independent founding and the determinate colony cycle, we have most ants and most termites. There are very few species that have this combination up here. No one is quite sure, and that brings me to a question. Let's take a look at the relationship between our thinking, our understanding of the life cycles of individuals and colony cycles of social insects. There is now a large, very mature body of knowledge and theory about life cycles. Lots of life cycle theory. It's a big topic. That on colony cycles lags far behind. Now you might expect that people studying colony cycles would take a hint from those studying life cycles and would draw predictions by analogy. You'd be right about that. Very sensible thing to do. Let me introduce a note of caution here. Sometimes people studying colonies and cycles will look at it and say, well, in life cycles this happens, therefore analogously in colony cycles this must happen. Well, no, it mustn't. Maybe it does. It's a good conjecture. But that's not the same as knowing that it does. And that's been one of my dissatisfactions with the biology of colony cycles. Now, one thing that they share is a rock-bottom fundamental concept of trade-offs, also in your glossary. Now, trade-off in biology means the same thing as it does in ordinary life. If you've got $100 and you need two things, each costing $100, you're going to get one of them but not the other. It's a trade-off. Trade-offs are what happens in ordinary life, as in biology, when you can't have everything. And an obvious example of a trade-off in life cycles and in colony cycles has to do with the size of the propagules. So you can think of some palms, for instance, such as coconut palms, that produce a whacking big seed. And that means that that seed has a much better chance of getting a good start. It's got a much better chance of successfully turning into a plant. And there are some other ones, such as the cocoa reed palm, for instance, that produce, they may produce the same mass of seed altogether, but they produce lots and lots of little ones. So there's a possibility that they might be much more successful reproductively. But each of those individual seeds has less of a chance, is less likely. And you think likewise of some animals that invest a lot in a small number of offspring versus investing just a little and none at all in a whole lot of offspring. So that's the kind of trade-off that happens in life cycles, and it also happens in these. So an obvious example of it happening there is in the distinction between independent founding and swarm founding. So a honeybee colony, for instance, has, when it swarms, and it divides, let's say it divides into two equal size smaller colonies. Well, each of those has a good chance of success, but the number of propagules is very small. On the other hand, Jack Spaniard wasps can potentially produce a lot more new colonies, but you would expect the failure rate of new colonies to be much higher. And you'd be right about that. Okay. Now, for most of the rest of my time, what I would like to do is to report results of one of the bug group's research projects. It's the most productive of our research projects to date. And let's go back. I think I've got, yeah, okay. Here's the Callistes colony cycle again. And the species we have been studying is the largest of our familiar Jack Spaniards, sort of mostly reddish, a little bit of yellow. It's Callistes laneo. And Callistes laneo is fairly abundant. And so we knew in advance that this was a diagram of Callistes laneo's colony cycle. So we knew the sequence of events. But that's not the same as knowing the pace at which these things unfold. It's not the same as telling us such things as the relative growth and survivorship in the different stages. So, for instance, and that applies to other animals, other organisms as well. So, for instance, you take sharks. Sharks all have pretty much the same life cycle. But that doesn't mean that the details may differ in a very significant way. So, for example, the great white sharks. Great white sharks, we're now just coming to understand, only start reproducing at about 20 years of age. And they may live to 70 years of age. They're among the longest lived of all animals. Well, that's an important part of what white sharks are all about. It's an important part of understanding the strategy of their life cycle. So the bug group undertook to analyze the colony cycle of Callistes laneo. And let's take a look at some pictures. So here's one in the founding stage. There's an incipient colony with just one queen. Could have more than one queen. And you can see that she's just got a small number of cells there. You can see an egg in that one. Very small nest. Is certainly in the founding stage. Now let's go to the next slide, please. And here you see one in the growth stage. And there you can see some pupal caps up there. But, some of those you can't tell from here, but some of those cells up at the top, some of the earliest cells, have already produced new individuals. So this one has passed beyond the founding stage and is now in the growth stage. So some of those individuals that you see there are workers. They are daughters of the colony. And if the colony is successful, it will, yes please, it will go on. And here is one in the reproductive stage. And you see it's much bigger. It's producing a lot more. Just look at the huge number of brood. This is a very successful colony. Now, do most colonies, here's a question that exercises. Do most colonies succeed in finally reproducing? In other words, do they mostly reach the reproductive stage? Or is the next generation of Ballistes laneo in that population mostly derived from just a very small minority of the colonies? That's I'll tell you what, I'm going to come back to that. Now, as Hans has pointed out, yes please, the old nests tend to persist for a long time. So these are nests of colonies that have died. There are different species. And so right away, this makes the point that you can, if you know the wasps in your area, you can almost always, with confidence, tell which species a particular nest is. That was critical to us undertaking this project, because what we did is we went first through the university field set station, and we collected each and every old nest that we could reach. Collected absolutely every single one of them. And then later we did a similar thing in Kernaham Village in Nariva. And we looked at it, and we could tell at what size a colony had died. Because once the colony dies, of course, it doesn't add any new cells, and it doesn't reduce cells as it goes along. So a colony can either remain at that size or it can get bigger. And there is, as you would expect, a very strong positive correlation between colony size and nest size. So the nest, this inert object that persists for years and years and years and it's under shelter, with very little degradation, is the nest is a good index of the final colony life. Now, I reported to you. No, let's go. Let's take a look inside a nest. So here are some larvae. So we could tell what size and approximately what age each of these nests reached, and we could get the frequency distribution. Now here's a series of life stages of wasp larvae. Why they switch them in between, I don't know. They don't do that. That was just a graphic error. Now, so you see an egg, 5 stages of larvae, and then this last stage undergoes a sort of a transformation just before pupating. And, yes please, Benoit? Yeah. Now look up at the top of that pupa, and there you see what's called a meconium or a fecal pellet. So just, they do not defecate all through larval life. And then just as they're about to pupate, they defecate all at once, and they lay in the top of the cell a nice, tight feces. Right there in the top. And then they spin a cocoon, which is outside of that. And then, let's imagine that this one emerges, comes out, and a new egg is laid, and a new larva, and it goes in there. When that one reaches that stage, it will lay a new one, it will put in a new fecal pellet. And these are distinct enough that you can go through, you can open up a cell, and you can count the fecal pellets. Seldom more than 3 in any cell. Very often just 1 or 2, or 0. And so you can know, it can be known by the simple drudgery of counting fecal pellets, how many mature individuals that colony has produced. So, let's see, where are we? Now let's go to, yes please, Benoit. Let's go to survivorship curves. Now what this shows is, here is the age, and these curves, these 3 idealized curves, represent the amount of survivorship. So a type 2 curve in the middle means that the chance of dying is equal at all ages. So there's a constant chance of dying. And some kinds of birds, for instance, have a fairly constant probability of dying throughout their life. There are many, such as this oak, that have extremely high mortality early on, but if an oak gets established, like this one here, it's probably going to continue to grow to be a very old oak. And humans were, through most of our history, were something like that. So that infant mortality was extremely high. I mean, you're well aware, I think, that in many societies, people didn't bother to name children until they were about 2 years old, because chances are no name was going to be needed. The kid would probably not survive. And then if the child did survive, it would probably reach 35 or 40. Okay? But through medical advances, at least in advanced industrialized countries, infant mortality is much less. So that now, in our society, among others, we have something closer to a type 1 survivorship curve. So that means that that newborn there will probably survive to adulthood. The mortality of children in advanced industrialized countries with good medical care is not very high. But then you get out there and you get to 60 years old, and the probability that you're going to die soon, Elijah and Hans are getting kind of nervous when I say that. I'm past 60 myself, so I better hurry up. I don't know if I'll make it through this. Okay? So those are the three idealized curves. Now, and this can be applied to colonies as well. Now, it had long been suggested that Jack Spaniard colonies typically had something close to a type 3 curve. That that mortality was very, very high in the founding stage, but then after that they were pretty well defended and would probably live out a normal lifespan. Now, if this looks familiar to you, that's because I've shown it to you before. I reported to you before on this initial phase of the Bug Groups project. And so we took more than something over 700 of these old nests. And we looked at nest size here in cells. And we looked at how many there were. So initially, in the beginning, there were something over 700. How many of them made it to 10 to 20 cells? Still a lot, but not as many. How many made it to 21 to 30 cells? Fewer, etc. Okay, and then it comes down. And then after 300 cells, they're practically none. Okay? So this is like a survivorship curve. Now, the point to which that I drew to your attention earlier, that I reported about a year ago, was that it looks like there's something of an inflection around here. It looks like it. Well, on average, the transition from the founding stage to the growth stage, remember that's when the first workers appear, comes around 33 to 34 cells. So this looks like it is in corroboration of that. However, notice that this is not absolute age. This is nest size. So what if nest growth is very slow in this place and speeds up out here? Well, what that would mean was that in time, this would be compacted, and it might appear more like a type, type 2 life cycle. It could be, but we weren't sure about that. So we said, okay, we've done the easy part. I mean, in fact, the initial part that produced this graph from the University Field Station, a similar one with some hundreds of nests from Kernahan, took us maybe half a week altogether. So we said, okay, now what we need to do is we need to monitor them and see how fast they grow. So we undertook the drudgery of monthly censuses in Kernahan Village. Why not the University Field Station? Much more convenient. Well, the reason was because the population there underwent a sudden drastic crash. Nobody knows why. I checked with the director. He said they hadn't gone around spreading them. It underwent a crash. And so we had to look around to find a new study site. We went to Trinidad Village. Well, and there we undertook monthly censuses. We had intended to do that for a year, 12 months. But then after 9 months, that population crashed. And once again, we don't know why. There's no evident reason for it. But we still have pretty good data. So one thing we learned there is that the colony cycle one thing we learned is that the populations of Polistes lano appear to be subject to occasional large crashes. And bear in mind, what this suggests is you've got a population crash, and then you've got possibly a genetic bottleneck. And that's not part of our study, but it's an interesting thing to bear in mind with respect to the genetic variability within the species. We also learned that the colony cycle was shorter than expected. Now, most of the research of these wasps of this genus has been done in North America, where the colony cycle lasts a year. While here, they are much shorter. So even that great big one I showed you a little while ago, that one is almost certainly less than 6 months old. We had very few that lasted as long as 6 months. So, is nest growth constant, or does it pick up over time? Well, as you would expect, bigger nests with more workers to do the building, on the whole, have faster growth. There's more cell building going on. That's expected. And what we expected to find, what we predicted in fact, was that right here, at this inflection point, when the first new workers emerge, that there would be a rapid increase in colony, in nest growth. Pretty sensible prediction. That's probably, if you were thinking about it just now, that's probably the same prediction you made. However, we didn't get anything of the sort. There is a rapid increase in nest growth, but it happens much later. There's a lag time, about equivalent in time to the founding stage. So that's what our data show. In addition, I hope I've got the right slide here. Yes, please, Mark. Okay. My student Sinead Stewart did a more finely grained study of a group of this one right here. A group of 34 founding stage colonies and 18 early growth stage colonies. And she went and she looked at them 3 times a week and counted them 3 times a week, not just once a month, as a bug group was doing. And if you just eyeball these data, it looks like there's not really any difference on average. And in fact, the mean nest growth per day is just about the same in the 2 groups. So you're going to get faster nest growth later in the cycle, but it'll be much later. Now, conducting monthly censuses allowed us to complete the data set in yet another way. So let's go back to that in the next forward, please. Okay, notice that this is nothing but old nests. You can think of ones that died of natural causes. And many colonies do go under for that reason. The nest is still there. But many are taken out by predators, especially birds, and when that happens, the whole nest is gone. So our monthly censuses allowed us also to look at whether there are many that come to the end of their days and just disappear, or maybe are found down on the ground. And in fact, the fraction is fairly high. So that requires some adjustment in this. Well, the question arises, is the average age of those that just die of what I'll call natural causes that just die, is that the same as those that get knocked off? And the answer is no. That these ones tend to be older. Ones that get knocked off just disappear, presumably knocked off by birds. We don't know of any other way that they could be gone. Tend to be younger. And I think you can see why. Younger colonies or smaller colonies, if you're going to go out and you're going to tangle with a wasp colony, and if you're a predator, you're not interested in the adults. You're interested in the brood. The brood is juicy. I've tasted them both. Brood is far superior. And the adults, they're just not meaty. They're kind of gristly. Okay? I recommend that the brood, you toast it lightly on a baking tin and just salt it lightly. Really.
You eat the legs only?
No, they don't have legs. You've seen the larvae. Okay, so it makes sense from the predator's point of view to tangle with the smaller ones that may have a significant amount of brood but have less there. So this is consistent with our working hypothesis that the main benefit of getting past the bounding stage, this very hazardous early stage, surviving infancy, you might say, and moving into the growth stage is not in being able to be more productive right away, but in being able to survive. Now, okay, we're doing well. It's just about time to wrap up. Now, okay, I'm skipping over a couple of things here. Okay. So our present estimate, coming back to the question of whether the next generation is the offspring, whether everybody reproduces or many colonies reproduce or just a small number of them, our present estimate, which is not bad, is that about half of the new colonies, about half of the adults that are produced come from just 10% of the colonies, the largest 10% of the colonies. And if that's the case, then in fact much more than half of the colony propagates come from just 10%. So that there is huge variance in the reproductive success of these colonies. So there are the members of the bug group. We were also were assisted by Palash and Stephen, and Suzanne Stewart is thanked for her contribution. Okay, I think we're ready for questions if I can find the lights.